Thank you very much for having me, giving me the opportunity to speak to you about our findings. You will see that they developed in a kind of totally different way than do you see the whole screen? Something? Oh, now it's there.
That looks great.
Yeah, now it's the start. I'm coming from a totally different corner. I started with a fungus that's used in industry that's been discovered many years ago during the Second World War. And from there, it developed like into the most important fungus for cellulase gene production and for heterologous proteins, so very, very industrial. And yeah, if you look at the whole situation, such a big fermenter is also just an environment for the fungus, and it's still doing or feeling what it feels in the environment. And so I always try to understand how does that work, because only if we understand, then we can improve it. So you see here the environment the fungus is coming from. It's a tropical environment. And in my case, the first thing I found during my PhD already, which was disappointing at the time, but we found light regulation. So enzymes are regulated by light. So I found it very interesting, this connection to plants sensing light, fungi sensing light. And it was difficult to spread the word, you have to be careful with light because it will mess up all your studies if you're not considering that light is influencing the whole pathway from the nutrient sensing to even the conformation of proteins that are landing on the promoter and of course the enzymes that are the outcome. So this was all very important, and we wanted to know the signal perception is so strong that in darkness you have the full capacity, but in light it almost drops to nothing. So there must be some regulation behind it. And what we tried to see is we used cellulose very close to nature, and then glucose, glycerol, and all that in light and darkness. So we had carbon sources that are inducing cellulases, and we had some that are repressing cellulases, so which in the world of the fungus means inducing means you need cellulases, you need to produce, you invest your energy to degrade whatever is out there. And if you have glucose, you don't care about that because all the buffet is there, the cake and so on, and you just eat. And so this is a very important thing for the fungus in terms of investment. And we were curious how that works from the beginning. And this is also a glimpse at the cascade that I will show you afterwards. So in the outside, you have sensors, G protein coupled receptors. Then you have the cascade with G protein alpha subunits, which are transmitting the signal, interacting with beta gamma subunits, then CIMP pathway, and all those together regulate cellulase gene expression. And what you then see with this northern plots is that GNA3, for example, if you constitutively activate GNA3, the signal transmitter, that is GNA3QL in this area, you get a very strong signal, but only in light. And then if you go for GNA1, the situation is different, but also light dependent. But what did not happen, what we expected, is if you constitutively activate the signal, so the pathway firing all the time, you don't get the cellulose all the time. So you're not telling the fungus, oh, there is cellulose out there, you need it all the time. And it didn't work. So this was disappointing at the time, but it totally makes sense. And that's what we found out, which was very interesting for us. And one of the outcomes of this story, checking all the different carbon sources and light and darkness via transcriptome, was we found many different G protein coupled receptors that could play a role in that because they were regulated in different comparisons, and we selected 4 of them. So what we see here is the most interesting one was CSG1, which is the representative of a class that was not described in Trichoderma so far. And when we first looked at CPH1 transcripts, well, nothing much happening. But when we looked at the activity levels, they totally dropped down. And then we went to lactose. So lactose is a special thing in Trichoderma ricei, because on lactose, cellulases are produced, even though the fungus doesn't need the cellulases to survive. So the interesting thing is lactose induces cellulases, even though the fungus, the degradation of lactose works in a different way without the cellulases. So it's good if you see here, there are no cellulases produced, would mean fungus cannot grow anyway. But this is not happening. And here we see this drop as well. So the conclusion was CSG1 senses something. At the time, we were not sure yet. But whatever it is, this signal works at the post-transcriptional level and induces the production of cellulases, of the proteins that degrade. And then we were curious, what is it sensing? Yeah? So it was not a surface that was clear very early, but then we tried glucose because, you know, the overlap between lactose and cellulose is glucose. And what we found is a concentration dependent sensing. So how did we do that? We used chemotropic responses. That means you pour an agar with all the spores in it, and then on the one side you add a control, water for example, and on the other side you add your chemotropic agent. You wait for 13 hours and then you check where the spores are orienting. And that's how we found this concentration sensitivity. If you then delete either CSG1 or CSG2, they are the same group of class 13, and then this response is abolished. So that means we have found two glucose sensors, and one of them is responsible for switching on cellulases. And we were, of course, interested if you look at the whole transcriptome, what happens if you switch off this glucose sensing that is so important later on. And we also have transcriptome data from several other proteins. So, and the interesting thing was this pattern from the G protein coupled receptor. So that's a small part of the whole transcriptome. That is associated with YPR2, and this is a regulator of secondary metabolite genes. Additionally, we found that in darkness, in CSG1, the genes upregulated are enriched in function of secondary metabolism. And then I checked the networks with network inference to see where are the most important regulators that we should look at. And there was one, GPR16, that where here you see the arrow blinking. This one seems to be quite interesting, and it's abolished in the CSG1 transcriptome. So this should be an important one. And what does it do? We deleted it. And what we saw is, on the one hand, cellulases are gone in this GPR16. Next thing is it has an increased level of secondary metabolites. And on the other side, where you see the plates, there you can see that it kind of inhibits itself. So it produces obviously such a high amount of secondary metabolites that it decreases its own growth. And what is not shown here, just for space reasons, is that if you switch off the SOR cluster, then this response doesn't happen anymore. So it seems to be, if you have GPR16, it switches, if you delete GPR16, you induce a feedback that works via the SOR cluster. So you have a secondary metabolite like program that is switched on. Normally in the wild type, it's balanced. So with enzymes and also here, so that this all has some good levels. If you delete this sensor, then it goes out of the way. And what does it mean? Is it something like quorum sensing? And we go on then to see where does this pathway go. So first, we were looking at the level of signal reception, and then we asked, does it go through this cascade of GNA1, GNA2, GNA3? And actually, this is indeed the case. So with the deletion mutants, we didn't have the response anymore in PKIC, which is down the line a little bit, and PHLP is a phosphocene that interacts with beta and gamma, but itself seems to be not so important for this response. And then we asked, thinking of we come now to the natural habitat of trichoderma, because that's, in a way, this is interesting also for industry. That's also why we look, but it's much more interesting if you think of the background of the habitat. So if a fungus growing in nature, then it encounters debris, plant debris, dead material and degradation products of this material, like cellobios, pectin, and so on. And it needs to feed through all that to get through its glucose and cellulose. But the only thing that really showed a nice response was again glucose. So it seems to be glucose that represents plant material outside of the cell. And thinking of glucose, that's back to the thing, is it glucose, is it the cake, or is it something where you need to produce these energy intensive enzymes? So you need this balance, and this is done by CRE1, by carbon catabolite repression. And it's of course interesting if glucose is so important. What role does this carbon catabolite repression play? And we looked at our concentration dependence, and the first thing was, okay, concentration dependence is abolished at 1%. We didn't get a response anymore. But then we tried higher concentrations, and we see that it's not abolished, it's just shifted. And that's so funny, because in the early days, people were trying to improve their yield in fermentations. And what we saw, if you delete CRE1, then you get a higher yield. So usually Trichoderma is producing lots of cellulases with relatively low nutrients, which is clear because it needs this concentration, and this is relatively low. Deleting CRE1 gives you a higher concentration for the sensing. So this perfectly fits. And then we asked, well, in the environment, there are also living plants. What happens? Does Trichoderma interact with these living plants where there it could get the glucose? And indeed it does. So on top, you see a very good colonizer. It's a biocontrol fungus and QM6A. So this is our pet, the industrial grandfather kind of. And we see that if you delete CSG1 and CSG2, these two glucose sensors, you don't get the colonization anymore. So something is wrong with the communication or with the detection. And then we looked at dead plant matter. Can it still sense it? And what we saw is in the wild type, in these Heifer tips, relatively early in the growth, so not long after the germination, you see this broadened hyphae when they grow on the growth front. So my student called that the gecko feet of Trichoderma. And in the mutants, you don't see that. So it's normal Heifer tips, and they are obviously not attaching or something, or probably not feeling what is there. So there is an interaction between plant and the fungus that depends on glucose, and that is not there anymore. And we also tested if this chemotropic response to a root extract, so to something that the plant's exudate is still there. And also that was not there, also not to CRE1. So there it seems that the concentration is not fine anymore. So the fungus senses, okay, this is not a plant, something is wrong. And then another study came into our mind that was from the group of Antonio Di Pietro in Cordoba, and they had found that pheromone receptors are important for fungal plant sensing in Fusarium. What they didn't have in Fusarium, so this is an asexual fungus, and Trichoderma ricei is sexual. So of course we were very curious, if these sensors are important for us as well, and how important is sexual development anyway. So what we saw is if you delete the pheromone sensors, you don't get a response to the plant anymore. So fits perfectly. It's a conserved pathway, obviously. And then we tried the thing with the soybeans. What do fungi do if they encounter a plant when they are just having sexual development, meeting their counterpart? And what we saw is they still have sexual development. It's 1 day faster. So it's from 7 to 6 days, so not too much, but still 1 day is 1 day. And the roots, the fungi are sitting on the roots kind of and closing them in, and they look morphologically a little bit different. And so this picture is my absolute favorite, because you see that the fungus is kind of greeting the plant root. So there is something in this plant root that is really interesting for the fungus. And what we also see in the pictures with the staining, it's a live staining, that there is some interaction. So the plant is a little bit damaged, but the fungus as well. So it seems to be relevant, I wouldn't say important, but relevant for the fungus, that this is a carbon source, but the root is not killed. So they are kind of sitting on each other and, well, kind of discussing, but you don't harm me, I don't harm you. And it seems to be a good interaction. And this acceleration of sexual development, of the fruiting body formation, doesn't happen just with an extract. So you really need the plant there. So this means there must be an active communication. Otherwise, yeah, the fruiting bodies are formed, but not faster. And so as a summary of the whole story, what we found new is that, yes, you have an induction cycle for enzyme production via sopherose, but this happens only at the transcriptional level. When we delete our CSG1 at the transcriptional level, we still get the induction. So the fungus is preparing for whatever is out there, because the sopherose tells the fungus, okay, this is the right degradation product. I might need enzymes. But then it is also testing if I produce, so this is now the interpretation, if I produce enzymes, do I get the right amount of degradation products back? Do I really need the enzymes, or is there somewhere glucose lying around that I might eat anyway? And such sugars are likely to come from a plant. So we are quite sure it's not glucose itself or not glucose alone, but glucose is one important part in a concentration dependent manner that tells the fungus, okay, there is something out there I need to degrade. If you go down the pathway, we have an important checkpoint at Q1, because this is important for setting this concentration. And then we are also including the mating partner. So this tells us there is a shift between communication as well, between nutrient sensing, feeding, and development. So for the fungus, the interesting thing with all these different topics is the fungus needs to survive. It has a certain amount of energy that comes from the nutrients, and it needs to distribute them wisely. So, and the system, all these interdependencies, they tell us that, okay, I wouldn't say it's a brain, but it's balanced very well. So I don't sense glucose, then I need to shift the communication. I might fend off some competitors, or if there is a plant, I'm faster with the sexual development, so they adapt better to use the plant better. So this was a fascinating network that we are still working on, and I hope you are as excited as I am about the data. And with that, I want to thank all my students who did a wonderful job, because every little bar is counting 800 little spores, and we did that over and over again, and collaboration partners and the funding. And yeah, thank you very much for your attention. I'm almost in time.
Thank you.